Hello everyone, a very warm welcome to my plant physiology class. Today I will quickly revise C2 and C3 pathways or cycles with you. This is a revision session. If you have not gone through the original elaborate video lectures, please go through them. I'll share the links in the description box. So let's get started. So we will start with C2 cycle. C2 cycle, you all know, is otherwise known as photorespiration, or it is also known as oxidative photosynthetic carbon cycle. The two names are important, should not get confused. One is photorespiration, the other is oxidative photosynthetic carbon cycle. Oxidative photosynthetic carbon cycle. Okay. So as we all know, this is a wasteful process. Why do we say that it is a wasteful process? Because carbon gets wasted here. 75% of carbon gets recovered and 25% gets lost. Okay. So it's a energy wasting process that occur in C3 plants. Okay. Where does it occur? You must know. It occurs in C3 plants and at elevated temperature. That means at very high temperatures. You must be knowing names of some C3 plants. Can you name some of C3, some of the very well-known C3 plants? These are C3 plants means these are cereals, barley, rice, wheat, all these, huh? Rice, wheat, barley, sunflower, potato, tobacco. Right, etc. Okay. So let's come to the pathway. What happens here? Here I have already drawn it out. I've written it also for your convenience. So here you see it occurs in three organelles. That means it involves three organelles: the mitochondria, peroxisome, and chloroplast. Okay. See here, ribulose 1,5-bisphosphate is the starting molecule. If you just consider it like this. So RUBP in association with oxygen forms 2-phosphoglycolate, the reaction being mediated by rubisco. Here, oxygenase activity of rubisco generates, see here, oxygenase, remember this is the most important key point here. Oxygenase activity of rubisco generates one molecule each. One molecule each of phosphoglyceric acid, PGA, which is a 3-carbon compound, and phosphoglycolate. Phosphoglycolate, which is a 2-carbon compound. Fine. So here you see RUBP is forming 2-phosphoglycolate. 2-phosphoglycolate with the help of the enzyme Rubisco. This phosphoglycolate is then getting converted to glycolate. This phosphoglycolate is then getting converted to glycolate with the help of the enzyme phosphoglycolate phosphatase. Phosphoglycolate phosphatase. What is happening here? See, inorganic phosphate goes out and phosphoglycolate phosphatase forms glycolate from phosphoglycolate. This glycolate arrives in the peroxisome where it forms glyoxylate with the help of the enzyme glycolate oxidase. Here, which enzyme comes into play? Glycolate oxidase. Glycolate oxidase forms glyoxylate here. Okay. And here you see this H2O2 that is formed here. This H2O2 is reacted upon by peroxidase enzyme. This H2O2, this is reacted upon by peroxidase enzyme. Otherwise, what will happen? This will create oxidative stress in the plant. Fine. Now, this glyoxylate will form glycine. This glyoxylate then forms glycine with the help of the enzyme glutamate glyoxylate aminotransferase. What is the enzyme here? Glutamate glyoxylate aminotransferase. This is the enzyme which forms glycine. This glycine then gets converted to, or it forms serine. Here you will have to take a note of certain reactions. Here you see CO2 goes out. This goes out. And NAD is forming NADH plus H plus. NADH plus H plus. This is getting reduced. And which is the key enzyme here? Glycine decarboxylase complex. Glycine decarboxylase complex. What is happening here? See, this carbon is going out. This is that 25% of carbon that is lost. This is lost. That is not recovered. Clear? Now what will happen to this serine? The serine goes into the peroxisome. This glycine to serine, this reaction occurs in mitochondria, and this serine from mitochondria goes into peroxisome through a translocator. This is known as amino acid translocator. This translocator is known as amino acid translocator. Clear? This is known as amino acid translocator. Now this serine is forming hydroxypyruvate with the help of the enzyme serine glyoxylate aminotransferase. Okay, serine, writing here, serine glyoxylate aminotransferase. Okay, serine glyoxylate aminotransferase. Fine. Now this hydroxypyruvate will form glycerate, where again, see this reaction, this NADH plus H plus is getting converted to NAD. Fine. This is mediated by the enzyme hydroxypyruvate reductase. Hydroxypyruvate reductase. Okay. 1 second. I'm writing the enzyme here. Hydroxypyruvate reductase. Okay. This is forming what? Glycerate. Clear? This glycerate is now moving into the chloroplast through another translocator, which is known as glycerate glycolate translocator. Okay. Glycerate glycolate translocator. Remember the names, glycerate glycolate translocator. Okay, this is one translocator. This is the other translocator. What is the name? Glycerate glycolate translocator. Now this glycerate will form PGA. Fine, using ATP. This glycerate will form PGA. Fine, PGA means phosphoglyceric acid with the help of the enzyme what? Glycerate kinase. Glycerate kinase. Now this PGA will again regenerate ribulose 1,5-bisphosphate through the Kelvin cycle. Clear? So this is the entire picture of photorespiration. This is the entire picture of photorespiration. Clear? So what is basically happening here? The glycolate pathway is recovering 75% of carbon of photorespiratory product, while 25% carbon are lost in the form of CO2. This has to be kept in mind. Okay. Now we will quickly move on to C3 cycle. C3 cycle is something which you already know. It is nothing new to you. See, these are certain important things which keeps on coming in every exam. Okay, so what is C3 cycle? This is also known as Kelvin cycle, right? The Kelvin cycle reactions, as we all know, is a series of biochemical redox reaction that takes place in the stroma. You must be knowing Kelvin cycle reactions takes place in the stroma of the chloroplast, right? Now this, okay, let's write the other name. Kelvin cycle. The reactions of Kelvin cycle can be divided into three main stages. Carbon fixation. These little things you know, carbon fixation, reduction, and regeneration of the starting molecule. So what happens in carbon fixation? Let's see what is happening here in carbon fixation. Three CO2 molecules will combine with three 5-carbon acceptor molecule, right? 3 CO2 combine with what? 3 carbon dioxide molecules combine with 3 5C carbon acceptor, right? 5C carbon acceptor. What is that? RUBP. Clear? This is that 5-carbon CO2 acceptor molecule. Ribulose 1,5-bisphosphate. Clear? Now, this step makes a 6-carbon compound that splits into 2 molecules of 3-carbon compound, that is 3-PGA. Therefore, what is happening? 6 molecules of 3-PGA are formed, which gives a net count of 18 carbons. You agree to the point? The enzyme involved here is Rubisco. Here we have enough carbon dioxide. So oxygenase activity is not seen here. Rubisco means what? RUBP, carboxylase oxygenase. This one. We don't get to see the oxygenase activity of Rubisco. We'll see the carboxylase activity here. So what's happening? See, let's write CO2 plus RUBP gives what? 3-PGA. This is what is happening actually. See here what is actually happening, you see. 3 5-carbon compound means what? 3 into 5 is equal to 15. 15 carbon with 3 CO2 is forming 18 carbon compound, right? 18 carbon means what? 18 carbon means 6 molecules of 3-PGA. 6 molecules of 3-PGA. That means 6 into 3, 18. Clear? Now we'll go to the reduction phase. Now the CO2 is added to the second carbon of RUBP. Remember, CO2 is added to the second carbon of RUBP. Keep this in mind. Next, we'll go to reduction phase. Now, what is getting reduced here? This 3-PGA is getting reduced to 1,3-bisphosphoglyceric acid or 1,3-bisphosphoglycerate. Huh? So in the second stage, ATP and NADPH are used to convert 3-PGA molecules into molecules of 3C sugar. Finally, you get that glyceraldehyde 3-phosphate. Finally, 6 molecules of glyceraldehyde 3-phosphates are formed. And in this stage, NADPH donates electron to or reduces a 3-carbon intermediate to make this G3P. So let's write what's happening here. See. This 3-PGA is forming what? See, it's forming 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate with the enzyme, see, phosphoglycerate kinase. I'm writing one here. It's a big name. Phosphoglycerate kinase. From this name kinase, you can get an idea that here we have something to do with ATP. See. ATP. So ATP is forming ADP. That means it's getting used. Right? Now see, this 1,3-bisphosphoglycerate is then getting converted to glyceraldehyde 3-phosphate. Fine. Now this one is forming glyceraldehyde 3-phosphate. Clear? This glyceraldehyde 3-phosphate generation is mediated by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Here I'll write 2. Glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. Okay, then what is happening here? NADPH dehydrogenation is happening. So NADPH is forming NADP plus. Clear? Nothing difficult here? Okay. Next, coming to this regeneration phase. Now, one molecule of G3P, this G3P, glyceraldehyde 3-phosphate, this one, this one molecule of G3P goes to make glucose. Okay. While the other 5 must be recycled. Pay attention. While the other 5, see 6 molecules were there, correct? 6 molecules of G3P. 6. Remember, sorry, 6 molecules of G3P. Mind it, 6 molecules are here. Out of this 6, 1 molecule goes to make glucose, while the other 5 molecules must be recycled to regenerate the RUBP acceptor. That is why we call it what? We call it regeneration phase. Okay. Regeneration requires ATP. Okay. Regeneration requires ATP and involves a complete network of reaction. See, try to understand the concept. In order for one G3P to exit the cycle, 3 CO2 molecules must enter the cycle. Fine. It will provide 3 new atoms of fixed carbon. Now, when 3 CO2 molecules enter the cycle, 6 G3P molecules are made. See here, we have started with 3 CO2 molecules, and finally we have ended with 6 molecules of G3P. One exits the cycle and is used to make glucose, while the other 5 are recycled to make 3 molecules of RUBP. 3 molecules of RUBP means what? RUBP is a 5 carbon compound. 3 molecules of RUBP means what? 3 into 5 equal to 15 carbon. Since RUBP is a 5 carbon compound, right? And here, see, 5 molecules of G3P are going. G3P is a 3 carbon compound. Here also you have, see, 5 molecules of G3P means what? 5 into 3, which is 15 carbon, because G3P is a 3 carbon compound. So, equation is telling. Now, you must remember 3 turns of Kelvin cycles are needed to make one G3P molecule that can exit the cycle and go towards making glucose. This you will have to remember. Fine. Now, if you talk about ATP now, 9 ATPs are converted to 9 ADPs. Fine. 6 during fixation step and 3 during regeneration step. And NADPH, if you see, 6 NADPH are converted to 6 NADP during the fixation. It can be considered as fixation only. This carbon fixation and reduction, they go hand in hand, correct? So, in 6 turns of cycle means what? 6 turns of cycle means 6 CO2, 18 ATP and 12 NADPH are used. 6 turns of the cycle, it means, see, 6 turns of the cycle means what? 6 CO2, 18 ATP, 12 NADPH. So this is the basic calculation. Correct? So one glucose, if you need to keep the summary of the story in mind, you will have to remember that one glucose formation involves 6 terms of Kelvin cycle. You will not forget this at all. Okay? Clear? So here we finish our quick revision of the two cycles, C2 and C3. We'll soon be back with quick revision of glycolysis. Till then, keep learning, keep revising, and above all, maintain good health so that you can do these things. Okay? So, see you soon. Till then, goodbye.